It's my great pleasure and honor to introduce Professor Hans-Peter Piepho, who I hope for this community for sure needs very little introduction. And if you're a young new student, I think this is a great opportunity to hear the wisdom of Hans-Peter for many years of experience. So I'll hand it over to you to get the next take on mixed models for incorporating environmental covariables. Hans-Peter.
Yeah, great. Thank you very much for the nice introductions. Great to be here. Great to see such a big crowd. I want to pick it up from where Fred started, kind of, from basic models. And my perspective is going to be prediction. And here I've depicted the target population of environments that you've heard a lot about already. So we have lots of trial locations, trial environments that represent, hopefully, that target population of environments. And then we have like a mean, like the average in that target population of environments. That's my filled dot there. And then there's a farm. So we're ultimately targeting the farm. So we want to make a prediction for the farm. And when we make predictions, what we have is the data from those trial locations. But those trial locations are never identical to the locations where the farms are at. And that makes prediction difficult and challenging. So that's going to be my perspective. And this is a quote from an old paper that a lot of you know who've worked on G by E. This is where Finlay-Wilkinson, it's called Finlay-Wilkinson regression, but this is really the starting point, a regression of genotype environment yields on the environmental mean. This has come from this paper. And they make the point that really to be able to have useful models, to have predictive models, we need to have ideally a random sample of environments. Our locations should be random. Now I know that it's really hard to have a truly random sample of environments, but I think in all the modeling that we do, and certainly when it comes to prediction, it is very useful to have that perspective where we regard environments as a random factor. And then if that's what we do, we could use that assumption in setting up the mixed model. And there's some basic principles there. If we use a mixed model, we need to first define the status of the factors involved, genotypes and environments, as either fixed or random. And then if a factor is random, then all the effects associated with that factor, or all those effects that comprise that factor, are random as well. So if environment is considered as a random factor, it follows that G by E is random. That for me is the main motivation for modeling G by E as random, regardless of whether or not genotypes are modeled as random or fixed. So here the basic ANOVA model that Fred showed also. So either we have just genotype and environment, or we factor the environments by locations and years, which I think is very useful and important. And then we have all the two-way interactions and the three-way interaction of genotype by location by year. So in what follows, I'm basically regarding locations and years as random factors. Genotypes as fixed or random, depending on the particular objective. So the philosophy here, it really is, in what I'm presenting, environments must be a random factor, genotypes can be random or can be fixed. So we have, as you know, all the different kinds of information that we want to integrate. We have the phenotypic data to begin with, and I'll start out with phenotypic data and then see how can we integrate environmental data. Genetic data can be integrated as well, of course, but I'll not delve into that too much. So here's an attempt to provide an overview of the types of model that we can use to bring in the environmental information, environmental covariates. And there's two basic kinds, I would say, depending on whether the factors involved are qualitative, categorical, or quantitative. So can we stratify the environments into zones or regions, or can we identify environmental types using the environmental covariate information? Then we have a qualitative level characterization of the environments, and there's models for that, which basically try to model the covariance between the environmental types or the covariance between zones in terms of the genotypes performances in those different categories. And the other kind of model is regression models, where we integrate the environmental covariates into regression models. And they come in at different levels of complexity. They come under different names, but my name for the most complex one is factorial regression, which is very old, and Fred's done a lot of work in that space with Jean-Baptiste Denis. At the other end of the spectrum, there's the environmental kinship approach, which in a way is the simplest model. And then in between, there's reduced rank regression and Finlay-Wilkinson regression and extensions of it. So what I want to try and do is focus on those regression types of models and tackle three issues. And the main one is, how are these models, these seemingly different models, how are they related? Well, as I've already pointed out in what I'm doing, I'll be factoring environments by locations as yes, that's going to be taken for granted. And towards the end, I'll talk about assessment of uncertainty. What is driving the uncertainty? And one major problem, I think, is the uncertainty about the environmental covariate values themselves. And I think that's sometimes neglected. So here's the basic factorial regression model, where you've got a response, eta ij for genotype I in environment j, and you've got an intercept alpha, and then you've got the slopes gamma times the covariates x. So that's the factorial regression model. We've got a separate regression, multiple linear regression for each of the genotypes. This is a pretty straightforward model. Now, how does that fit into the mixed model context? And if environments, how does it fit in particular when environments are random? Well, there needs to be random deviations from those regressions. And I'm showing this to have two effects there: a random main effect for the environment, U, and a random residual E. And both of those will be factored by years and locations as usual. And the residual then comprises unexplained parts of the genotype-environment interaction. And that makes it a mixed model right away. But now we're going to also look at genotype as a random factor, and how does that play out in this type of model. So we've got the intercepts and we've got the slopes. So if those are both going to be random, then we need a mean of all the intercepts across all the genotypes. This is my mu alpha. And we need a mean of our slopes across all the genotypes. That's the mu gammas. And then we have random deviations that are genotype-specific. So A is the random deviation for the intercept, and C is the random deviation for the slopes. And then we can plug it all into our model, and that makes it a random effects model on the side of the genotypes. And then we can look at the joint distribution of the intercepts and slopes, and then we have different choices. So for the three choices that I'll be looking into, we assume normality, zero mean, and some variance-covariance matrix sigma. And the models can be characterized in terms of the choice for sigma, the variance-covariance matrix between intercepts and slopes. But before we go there, another look at that same basic model. So we can partition it into a mean regression across the genotypes. That's the F at the top of the slide. And you have a genotype-specific deviation from that mean regression. That's my function G, which is both of them are functions of the covariates, and I've written them out here. And the key point there is that the deviations from the mean regression, the genotype-specific deviations, they are random because the intercepts and slopes are random as well. And we can also point out that we have an explicit model for the environmental main effect, which I call eta. That's my mean regression. So all the mean regression terms there. But then we have an added random effect that models the deviation from those from that average regression in each individual environment. And it's important to have that, to have a complete model, because we can't expect the regression to explain everything, of course. So now, when it comes to specifying the variance-covariance matrix for those intercepts and slopes, it's convenient to put all the covariates into a matrix X, as I've done here, and see how that translates into the variance-covariance matrix. So for our responses are the eta ij's, which I'm now collecting into a vector for each genotype. So eta I on the top of the slide is the expected values of genotype I in all the environments, given the covariates. So those have a normal distribution, a multivariate normal distribution as shown here, with a variance-covariance matrix, omega, which depends on the covariates, of course, and it also depends on the variance-covariance matrix, sigma, that we assume for the intercepts and slopes. And there's 3 different choices. One option is the unstructured model. And that, from a statistical point of view, is the most obvious choice. And it's very commonly used in random coefficients regression. This kind of model is known as random coefficients regression in other contexts. And why unstructured? So unstructured means each slope has its own variance, and each pair of slopes has its own covariance. And there's also a covariance between the intercept and the slope, each of the slopes. And for each, we have a different covariance. So that's a complex model, lots of parameters. But why is that necessary? Why is that a desirable thing to have if you can fit it, which is the challenge, is so you have what's known as translational invariance. So your model fit does not change when you do apply a linear transformation to your covariates. We want our fit to be unique no matter how we scale the covariates or in which units we measure the covariates and things like that. So that's the unstructured model, but it's also the most complex one. So it's most realistic in a way, but it's also costly. And then at the other end, we have the kinship approach or kernel approach by Jarkin, and sigma takes this form. And maybe you don't see it written out that more expressed in that way, but this makes a nice link with the unstructured model. It says there's no covariance between intercepts and slopes. And it also says that each slope is drawn from the same normal distribution. So we don't have that heterogeneity of variance between the different slopes corresponding to the different covariance. So it's a much simpler model. It's much more parsimonious, but you could also say it's much more simplistic. Right, and it can be written in different forms. You can translate that form of sigma, the variance, covariance, or intercept and slope, into the omega for the data, and then you have the x times x transpose, which is your kinship matrix, your environmental kinship matrix. Okay, and that's the way that it's usually fitted, and fitting it in that way is particularly advantageous if you have a large number of environmental covariates. Okay, so here it is. And then in between, there's reduced rank regression, where we say sigma is some reduced rank expression such as this one, lambda times lambda transpose. So the matrix lambda has loadings pertaining to factors for the different environments. And this can be a very good approximation to the unstructured model. And Daniel Tolhurst, who's in the audience as well, has worked on this quite a bit. If you use this model for sigma, you can rewrite the model in different ways. And one of the ways you can do that leads you to an expression which I think is useful. I'll skip this slide, just rewriting the gamma. So we have one bit, the small gamma alpha is pertaining to the intercepts, and then the lambda sub gamma pertains to the slopes. And that's what I'm going to put the focus on. So you can do a little bit of rearrangement, and you can rewrite the model, which is originally a regression on the observed environmental covariate X, as a model that involves a regression on a matrix Z. And in that matrix Z, what you have is linear combinations of the observed covariates. So each observed covariates times some coefficient lambda. So the coefficients in this linear combination come from that matrix lambda. And this this is a useful expression because we can actually regard this type of model to work with what can be called synthetic covariates. So instead of working with the individual observed covariates, we form a linear combination or linear combinations of the observed covariates. We just need to work out the coefficients. And how do we work out the coefficients? One way of doing that is fitting that factor-analytic or reduced rank model and extract those lambdas, which will give you the coefficients to use to come up with that synthetic covariate. And we've used that same kind of model to do just that, extract synthetic covariates and do regressions with those. And that's a simpler model than working with the original observed covariates. There's a host of other methods that are very similar in spirit and basically achieve the same thing. Partial least squares regression is doing basically the same thing, but it doesn't do that in a mixed model framework. It does that using ordinary least squares, basically. Right, I think I need to move and skip this slide. So this is the idea of what we call extended Finlay-Wilkinson regression. Fit a factor-analytic model for the covariance among the slopes. At that step, we actually modeled the intercept for genotypes as fixed, which made it simpler to fit the model, because the main purpose at that point was to extract the coefficients for our synthetic covariates and then compute those synthetic covariates Z and use those in a regression model. And that model can actually be one where you have a fixed effect for intercept and slope for the genotype as your final model if you want to. But then if you have marker information, you can bring that in, use genetic kinship and so forth. Right. So that's the kinds of models, and each of those can be used for making predictions. At the end here, I want to point out that I think we should also be thinking quite carefully about the environmental covariates, and in particular about a mixed model for the environmental covariates. We don't usually do that. We just take, okay, these are observed covariates, we can measure them, that's that. But I think there's good reason to have a good statistical model for the environmental covariates. Why? Because when it comes to prediction, we want to predict for a farm where maybe we haven't conducted any trials, or surely, quite likely haven't conducted any trials, so that's kind of random. We can have data on the covariates, all right, but what about if we want to make a prediction for next year? We can't. We can't know what the rainfall is going to be. So there's that uncertainty, and to deal with that, it's good to have a mixed model for the environmental covariates as well. And we can, and that's something we're currently working on, use a fitted mixed model, and from that fitted mixed model derive, quantify the uncertainty, the mean squared error of prediction. But to be able to do that, we need that model not only for the phenotypes, but also for the environmental covariates. And in a nutshell, the problem is we're working with these products, a slope times a covariate value, but then for that covariate value in the next year, we maybe need to plug in an expected value. So we need to think about the variance around that expected value for the observation that we draw for next year. And how does that translate into the mean squared error of prediction if we want to do it model-based? Okay, so a contribution to the prediction variance is something like this here. The gamma, the regression coefficient squared times the variance. That's why we need a model for the covariates. And then we also need to deal with the fact that we are actually estimating the regression coefficient, but we're also estimating a mean for the covariate. So we're dealing with two parameters that are in a product, and they're both estimated. So what's the variance of that is something to work on. And we can use equations like this, but they do require these models on both sides, for environments and for covariates. When it comes to prediction, we can consider different scenarios, but I think the most interesting, but also the most challenging one is a new location, an unseen location, and an unseen year. And I think we also need to think carefully about the way that we set up the cross-validation scheme if we want to do cross-validation. I mean, I just said if we have a method to derive the mean squared error from the fitted model, that's nice, but the most transparent way or maybe convincing way to assess the predictive accuracy is by cross-validation. But how exactly do we set that up? Very often, what we do is, okay, we leave out the phenotypic data, but then we use the environmental covariates from the left-out environment. But if it's the next year, a lot of those covariates, their value will not be known. That's where the challenge is. And oftentimes, the cross-validation schemes don't really reflect that challenge. And it really does make a difference. I am running out of time, so I need to skip. Here's an example, just showing one slide more where we have two cross-validation schemes. And CV1 is basically the simpler one that I described, and CV2 is where we really leave out the whole year, including the covariate information, and instead we plug in means across previous years. And all the metrics like the correlation or mean squared errors, they deteriorate from one to the other cross-validation scheme. So my message is we really need to think carefully about the way that we set up a cross-validation scheme, if that's what we do. And what I've briefly mentioned, this model-based assessment, we also need to take care of that and take that into consideration. And I think I'll just stop there. Thank you.
Thanks, Hans-Peter. I'll take one question, if anybody's got one. Yep, Daniel.
Very nice talk.
Oof.
Very nice talk. Thank you very much, Hans-Peter. I was just wondering about, at the start of the talk, you said about your trials being adjacent or not exactly where the farms are. And of course, there are field trials that are placed on farm. So I'm wondering about this whole idea about random environments versus fixed environments, but maybe not a fixed environment per se, but rather the fixed location and the random year, and how you can incorporate that into what you've basically your framework here, basically disentangling the G by Y by G by L components, and obviously overcomplicating things, but.
Yeah, that's a good point. I mean, year is definitely random. I think there's no discussion about that, but about location, we can argue, and if I am at a farm, then the things might look different. But I'm just thinking in terms of, where's the locations where we do the trials? Whether it's on farm or on station. Even if there are some farms, or maybe lots of farms, there will still be lots of farms not in the trials, and we still have that same problem. So how do we make that transition from where the trials are, whether it's farm or experimental station, to farms where we don't have trials? And that's where I think that random assumption is still useful, but maybe it becomes a mix of fixed and random, and we can modify the model to accommodate that setting. So I think on-farm is great if we have on-farm, because that's really where, in the end, we want to be. So we need to think about that as well. Thank you.
Thanks. Really good question, and I think it shows part of the enthusiasm for these symposia is trying to form some of these challenging questions that we really have to be thinking about, so bringing people together. Thanks, Daniel. I think we'll leave it there. Hans-Peter, I'm doing a bad job of keeping us on track.